Once again, you are being afflicted with Mark and Bob, the grumpy scientists. We've removed some of the grumpiness from our multimedia command and control center, but Mark, that's because Mark's going to draw some stuff.
Yeah.
Today what are we going to talk about today?
Well, not a cat. I mean, because, you know, we notice that if you type in grumpy science, the grumpy cat
always come to the
is
top,
always above us. There's no way we can ever top it.
I can't look at a grumpy cat without laughing. But today we are going to talk a little bit about recent news about the brain. And when we looked at the 20 top stories about neuroscience in 2017, what did we discover? That 8 of the 20 top neuroscience stories involved the gut or metabolism and the interaction between the gut and metabolism and the brain.
Yeah.
Very interesting. 8 out of 20. That's almost half.
Yeah. And the other thing, and basically we have several things to talk about, but we won't do all of them. But
or we might. It's hard to say.
Or we might start talking about one thing and get off track and just talk about something else.
So we think that's going to be the topic today.
So
we could be wrong.
Did you know what Alzheimer's is often referred to?
I do know what it's referred to as, and it's because you told me, as type 3 diabetes.
That's right. So people aren't aware. I mean, diabetes researchers for many years
metabolic
just talk about
disorder.
type 1 and type 2 diabetes, and maybe a few diabetes researchers still may not know that the brain has its own source, has cells that produce insulin.
I can tell you that last year when I met for the first time with a number of diabetes researchers at a major university, I asked them, what other tissue besides the pancreas, what other organ produces insulin? I knew this because Mark told me. I did not know that. I'm not a neuroscientist, but what other organ do you suppose produces its own insulin? The brain,
That's
right? So isn't that interesting? You get insulin from 2 and only 2, as far as we know, right?
Yeah.
Organs in the body, the brain and the pancreas. And so people who really know what the heck's going on with neurodegenerative diseases like Alzheimer's, they're looking at it maybe
wrong, but these diabetes researchers were not aware that cells in the brain produced insulin.
I asked them if they knew that. They were like, no, you're kidding me, really? Do you have any references? And that's what these people study is insulin.
First reported in 2011.
Well, okay, give them a break. There's many thousands.
And only a few years ago. Right. So there's only a couple of hundred references now to look at it.
But the fact is, it's really interesting if you think about it. To begin with, why should it be that the brain makes its own insulin? Of all the things that it needs to do, it doesn't need to be replicating the activity of some other organ, does it? So these are really, really interesting questions. And one of the things that Mark and I like to do is we are systems physiology people. So
took the words right out of my mouth,
as I often do. And so we try to look at an organism as
a system, and how. And basically, you know what the diving reflex is?
I sure do. Absolutely.
Maybe this would be good. And basically,
it's basic college physiology.
Thing, you go back to your old systems physiology. And in other words, when we say systems physiology, we mean we study the systems as a whole. How do the different parts interact? What are the big things? And one guy who is a real systems physiologist who died a few years ago, but I think he wrote 13 editions of his medical physiology book with a guy named Guyton.
Mm-hmm.
And, so he looked at things in terms of systems and how does, and if you're, but basically, why don't you explain what the diving reflex is?
Well, the diving reflex is, I know it's true for all mammals, and I think it may be true for all vertebrates. You would know the answer to that.
I think so.
But it's certainly true for all mammals. So what happens when the organism, even if it basically never gets in the water, like a cat who's never been in water will respond this way, right? So the diving reflex is all of these things that your body does as a system that seem to be coordinated by the reflex of having water in your mouth and not being able to breathe. It slows your heart rate down. It shunts circulation to critical areas like your brain, right? Mm-hmm. Changes your respiration drive and all kinds of other things as a survival mechanism, because man, you dove in the water, and it's something that's kind of fundamental, but it's really primitive, and it seems to be common across certainly all mammals.
And basically, it protects your brain at all costs.
At all costs. So your brain says, hey man, you're going to be diving in the water. You know what? I get all the oxygen, or almost all of it. I get all the really critical stuff, because if the brain doesn't work, then no sense
Believe
coming
it or
up out of the
not,
water.
you know, the brain works 24/7, like, well, most of your body parts do, but basically, it's always got to have a supply of insulin.
It always has to have a lot of things. I mean, your brain, I think, you can correct me if I'm wrong, but your brain is one of the body organs that really doesn't change a lot in its metabolic demands. It can regionally change. You can image those things with like fMRI and stuff like that. But overall, your brain is just a big power hog.
Yeah.
It's very expensive metabolically to have a brain. So, and it has to keep the power coming. Oh yeah, grumpy science.
I forgot to identify us, you know, because I was so eager to draw something. But
that is the multimedia command and control center. So we're going to talk about system physiology a little bit.
And so I basically just wanted to introduce the whole concept of what's going on in the brain in terms of metabolism. Well, in terms of what we know, minor knowledge about insulin producing cells, because I thought it's a pretty interesting thing. And basically, the brain, the cortex, which is one that we talked about as the main part of the brain that we want to study, it's got, Bob, do you know how many layers cortex has?
6.
6. So
I just guessed actually as
one at the top, 6 at the bottom. What's the Roman numeral for 6? That's VI. Gotcha. Okay, so and then you got IV for 4 or whatever. So we have 1 through 6. These are 4 layers, and basically these are you got cells
1 through 6 is 4.
Yeah. You know, 1, 4, 6, something like that. Okay, I didn't want to draw all the numbers and show off my handwriting too much. So we got a whole bunch of cells at the layer 4. Significance of this is that layer 4 is where you have inputs from the thalamus. So that's what that big arrow is. We got inputs from the thalamus. Well, what happens is, and you're probably wondering why we care about inputs to the thalamus. So we go from skin up the spinal cord to the thalamus, and then in the thalamus it goes into the layer 4 of the cortex. Then it goes, then after the inputs go in, then they spread out to these other areas over here, over all around. So anyway, input gets spread out through layer 4 and through everything else. Now, at the level layer 4, we have something called neurogliaform cells. Very specialized cells. They're inhibitory cells. We talked about excitation and inhibition, and basically inhibition turns off cells. If this input is extremely weak, you know what happens? Do you think you get any excitation here?
Guessing not.
Guessing not. Okay.
Maybe guessing so. I don't know. My guess is whatever is the right answer.
Well, you got it right then.
that is it. I actually don't know the answer to that.
When we turn on this particular cell, what happens is, with a very weak stimulus, it's a subthreshold stimulus. So subthreshold means you give a very weak stimulus, but it's not strong enough to feel.
Right.
Okay, so we give a stimulus that's not strong enough to feel, it actually does turn on some cells that turn off everything. And what that does, so the neurogliaform cells are actually inhibiting other cells in here, so that on the next stimulus,
you can maybe feel it. So is it sort of amplifying by lateral inhibition? Is that what's going on?
Well, they're actually it's called feed-forward inhibition. Okay. So feed-forward inhibition you think of as in terms of it's going to turn off whatever is ahead of it. And so you give a very weak stimulus, and you turn off these cells, and you keep turning off these cells, and you give another stimulus. Well, those cells are turned off, and the next stimulus is going to be even more effective on turning off the excitatory cells.
Feed-forward inhibition.
So we keep turning those cells off. Well, the interesting thing about feed-forward inhibition is that neurogliaform cells, which are what we call a particular type of inhibition, they're mediate GABA B mediated inhibition, and those cells
GABA B being a neurotransmitter.
That has to do with inhibition. There's three types of GABA that we know about. GABA A, GABA B, and GABA C. GABA C is only in the retina, so we really don't care about it. But GABA A and GABA B are two kinds of inhibitory neurotransmitters that we talk about, but or neurotransmitter mediated inhibition that we talk about. But basically, the importance of that is that it differentiates different types of inhibition, and it differentiates different types of drug applications that you might use if somebody's deficient in one thing or another. One thing that we found out through certain tests that tested feed-forward inhibition was that certain populations were very low in feed-forward inhibition. And then we came along, we found out that neurogliaform cells, which we suspected were responsible for that inhibition, so they're called neurogliaform cells, NGF. And so when they get excited, they were turning off another set of cells, and so that you couldn't feel anything until the excitation got so big it would feel it. Well, we looked at autism, and we looked at chronic pain, and all those populations had lower than normal feed-forward inhibition. And the way we tested it was we wanted to see well, how much feed-forward inhibition was there? We won't go into how we actually measured it, but
it's sciencey.
It's sciencey kind of
probably
crap.
a little too
But the interesting part was we said within 2011 that paper came out and said, oh, well, look, neurogliaform cells actually produce insulin, and actually there are places in the brain that have 10 to 100 times as much insulin as you need.
The plot thickens.
Or not as you need, but 10 to 100 times as much insulin as you find in the blood stream.
To keep this, to make this relevant, this is something to keep in mind, is that insulin is very potent, and it's highly controlled in your body, and it's if your brain's making it, it's not an accident, right?
No, not an accident. And there are feedback loops where when your brain's making insulin, it's also feeding back to a different part of the brain, which is telling your pancreas, I got plenty.
Right. So if your brain makes excess insulin, it says, look, I can't deal with too much. I've got too much already. It's going to tell somebody else to shut the tap off. And that means it's going to be telling your pancreas to kind of turn down the insulin, or vice versa. If
it's not enough,
it's going to say, hey, I need some more, right? And so
so you find all these insulin-related problems in a lot of neurological disorders, and it could be mediated by this. And so one thing we did was we had found populations where you had lower than normal feed-forward inhibition. So what's the logical thing to do? What's the logical group to test to see if the neurogliaform cell is in fact responsible for feed-forward inhibition? Would a population that has abnormally problematic insulin levels have a different feed-forward inhibition?
Type 2 diabetics? Maybe.
Exactly. We tested type 2 diabetes, and we found that group actually had 3 times the level of feed-forward inhibition as normal population.
Isn't this interesting? So what you have to understand is that insulin is viewed rightly by many as, the master of the endocrine, system for controlling, metabolic things. It sort of drives a lot of very other important hormones and things like that. So if your brain is really tuned in to the insulin level and everything, and it's using its own insulin level to modulate or suppress or change the pancreatic insulin, which is the insulin that's supplied to the rest of the body. Yeah, if you got a problem in your brain insulin, you could see how that could very easily relate to a somatic, body-wide insulin problem.
Exactly.
Just because there's a communication link. If the brain feels like it's totally starved of insulin, it's going to keep saying more, right? If it's way too much, it's going to keep saying less. Because like we talked about with the dive reflex, the brain likes to come first.
Exactly.
Right?
And so that's sort of central to some of the research that is ongoing now with the aging population, is that we want to see that because Alzheimer's, there's about an hour's worth of stuff to talk about, but we won't do that now.
We won't torture you that way. We say that for our students who are paying tens of thousands of dollars a year to be tortured by us. So.
Yeah. But anyway, this is something that we do with the brain gauges. We're actually measuring this, and then one of the actual things we want to do with pulse electromagnetic field is see, would it have an impact on this, because this is probably very sensitive to the kind of inflammation that
is brain inflammation,
maladaptation that's going on in a degenerative process that we think might get reversed by pulse electromagnetic field. So what we want to do is, as we just talked a little bit about TBI and pulse electromagnetic field studies that are ongoing, now we're talking about Alzheimer's type studies and pulse electromagnetic field studies that we'd like to do.
That we're talking about the 8 of the top 20 stories from 2017 on neuroscience related to the gut metabolic link between the brain and the body. And here we have a very clear potential mechanism that's not well understood yet. It was sort of first reported only 2011, right? So yeah, there's a real potential for doing things that modulate brain inflammation and other things that may cause a dysfunction in this system, and having that actually interact with the rest of the body at a metabolic level. Quite interesting.
So what we really want is to get viewer questions about this, so we don't make people suffer through a whole hour or two of what we're trying to.
Because I'm sure we're going to come back to this. So please, if you have a question, if you're interested in this, maybe dig out a paper or something, the pop literature that could be right, or wrong. Send us a comment to the email address that I will list below, and we will answer your questions.
That's the short version.
That's the short version.
You're welcome.
All right. So, thanks. You're very welcome. We're letting you go now. You can be happy. I think that's it, right?
That's it.
Thanks a lot.